All right, so get ready, folks, because today we're taking on a real medical mystery.
A real head-scratcher.
Yeah, we're talking malaria, but with a twist. Everyone knows it's a global health problem, right?
Right.
But it gets even scarier when you think about pregnant women.
Absolutely. It can have a devastating impact on pregnancies.
Yeah, we're talking increased risks of low birth weight, miscarriage.
And even stillbirth in some cases.
Exactly. And the culprit behind all this is that infected red blood cells, well, they basically glom onto the placenta.
Like little microscopic leeches.
Yeah. But the question is, how do they do that?
That's the million-dollar question, and thankfully, we've got some fresh insights from a brand new study. It was just published in the journal PLS Pathogens.
Oh, very timely then.
It is. It's called What Makes Malaria-Infected Cells Stick. So we're going to dive deep into that today.
Perfect. So it sounds like we've got our prime suspect, the sticky protein. It's called VRR2CSA.
That's the one. It's like the molecular Velcro that allows those infected red blood cells to attach to the placenta.
Velcro. Huh. Makes sense. So if scientists can figure out exactly how that Velcro works,
Then they might be able to find a way to block it, to stop it from binding.
and protect both mothers and babies.
Exactly.
Okay, I'm following so far. So we've got our sticky protein, VRSCSA, but how does it get so sticky in the first place? What makes it tick?
Well, that's where this new research comes in. They've uncovered a crucial player, an enzyme called CK2.
CK2.
It seems that this enzyme is essentially flipping the on switch for VR to CSA stickiness
Flipping the switch, huh? So how does that work?
through a process called phosphorylation. It's kind of like attaching a tiny little chemical tag to the protein.
Okay.
That tag changes the protein shape and makes it super grabby, basically.
Interesting. So we've got a sticky protein, VR2CSA, and this sneaky enzyme, CK2, that's modifying it. But couldn't there be other factors at play here, too? I mean, how can we be sure that CK2 is the main culprit?
That's a good question. And the researchers actually tackled that head on. They did some experiments where they specifically blocked CK2, the casein kinase 2 alpha subunit, to be precise.
Okay.
And when they did that, those infected cells suddenly lost their grip.
Like they'd been disarmed.
Exactly. But here's where things get a little more complicated, a little bit of a twist.
Oh.
Turns out there are actually two versions of CK2 that could be involved in this whole stickiness situation.
Two versions, yeah.
One version comes from our own human cells,
Okay.
and the other one is from the malaria parasite itself.
Hold on. So two versions of the same enzyme, both potentially making this protein sticky? It's getting complex now.
It is, a little bit. But what's really wild is that even though both versions of CK2 can phosphorylate VR2CSA, they don't work in exactly the same way.
Oh.
The human CK2, it's kind of a specialist, very picky about where it attaches that chemical tag.
Okay.
Whereas the parasite's version of the enzyme, it's more of a generalist. It'll modify multiple spots on VR2CSA.
So it's not just about whether the protein gets tagged, but also where it gets tagged, right? It's like molecular detective work at its finest.
Precisely. And to really get to the bottom of this, the researchers had to zero in on the most crucial spot, the prime target.
The bullseye.
You got it. And through a series of really cool techniques, they pinpointed that exact location. A single amino acid, a serine at position 1068 on VR2CSA.
Yep.
That's the spot that the human CK2 really likes to target. Remember how I said it was picky?
Yeah.
Well, that's its bullseye, serine 1068.
Got it. Serine 1068. But why is that one spot so important? Why all the fuss about this one amino acid?
Well, it turns out that this phosphorylation, this tagging process, it might not just be about making VR2CSA sticky.
Oh.
It might also play a role in getting the protein to the cell surface in the first place.
Ah. So it's like a 2-for-1 deal.
Kind of like that, yeah.
Yeah.
To test this theory, they basically created a modified version of VR2CSA where they swapped out that crucial serine. And guess what? The cells could still produce the protein, but way less of it actually made it to the surface.
To where it could cause trouble.
Exactly.
Fascinating. So by targeting this one tiny spot, serine 1068, we could potentially stop VR2CSA from even getting to the surface and prevent it from sticking to the placenta.
A double whammy.
It is a double whammy. But wait, hold on a second. If both our cells and the parasite have CK2, wouldn't blocking it mess with our own bodies too?
That's the challenge, isn't it? And that's what researchers are now trying to figure out. How do we disarm the parasite's CK2 without causing any collateral damage to our own cells?
It's a tough one. But if they can crack that, it could be a game changer in the fight against malaria, especially for pregnant women.
A real breakthrough. It really is amazing when you think about it, isn't it? how targeting one tiny little amino acid could potentially have such a huge impact on malaria.
It is. It really makes you appreciate just how powerful those tiny molecular players can be. But let's be real for a sec. We're talking about malaria here. I mean, this parasite's been around for centuries.
Oh, yeah.
Evolving, adapting. It's got to have some backup plans, some tricks up its sleeve. Right?
Oh, absolutely. Malaria parasites are masters of disguise, you could say. They've got a whole arsenal of tricks to evade our immune system and, well, just cause chaos.
Chaos. And one of their most impressive tricks is probably their ability to switch up their surface protein. That's right.
It is, yeah. It's like they're constantly changing their outfit so they can stay hidden from our defenses.
So they've got one outfit for hanging out in the mosquito, and then they change into another outfit when they're in our bloodstream.
Exactly.
And then maybe even another one for when they're sticking to the placenta.
You got it. It's a microscopic fashion show of survival.
I love it. A microscopic fashion show.
But of course, it makes it very hard to develop effective drugs and vaccines.
Right, because you don't know what you're aiming at half the time.
Exactly. And to make things even more complicated, different strains of malaria parasites have slightly different versions of those proteins as well.
So it's like there are different gangs of criminals out there,
Right.
each with their own special set of tools and tactics.
Yep, pretty much.
Okay, so we've got protein switching, we've got all these different strands. We haven't even talked about all the other proteins involved. This is starting to feel like Mission Impossible. How are we supposed to keep up with all of this?
Well, it is a challenge, no doubt. But the good news is that with every new study, every new piece of information, we get a little bit closer to understanding all these complex mechanisms and coming up with solutions.
So there's hope on the horizon. Give me some good news.
Oh, absolutely. I mean, despite the challenges, there have been some really significant advances in malaria research in recent years. We have a much better understanding now of the parasite's biology.
Right.
We understand the immune response to the infection better, and all those factors that contribute to drug resistance.
Yeah.
And this knowledge is really paving the way for new and innovative approaches.
Okay, I'm listening. What are some of the most promising avenues for future research? Where should we be focusing our efforts?
Well, one very promising area is the development of new drugs that target essential parasite pathways.
Okay.
So researchers are working on drugs that interfere with the parasite's ability to synthesize DNA,
Okay.
to make proteins, essential metabolites, things that it needs to survive.
All right, the essentials.
Exactly. And these drugs could effectively just shut down those essential functions and lead to the demise of the parasite.
Sounds promising. But what about drug resistance? It seems like every time we come up with a new drug, the parasite finds a way to outsmart it.
Drug resistance is a huge hurdle. There's no doubt about that.
Okay.
But it's not insurmountable. One approach is to develop combination therapies,
Okay.
So drugs that target multiple parasite pathways at the same time.
They hit them from all sides.
Exactly. This makes it much harder for the parasite to become resistant because it would need to acquire multiple mutations to survive.
Right, it's a much higher bar.
Exactly.
What are some other strategies for tackling this resistance problem?
Well, another very promising approach is to develop drugs that target the host factors.
Host factors,
Yeah.
meaning what exactly?
Well, so instead of going after the parasite directly,
Yeah.
what we're doing is we're going after things that the parasite needs to survive in our body.
Okay.
For example, researchers are looking at drugs that block the interaction between parasite proteins and human red blood cell proteins.
So it's like cutting off their supplies.
Exactly.
Instead of attacking them directly, we're cutting off their supply lines.
Yeah.
I like that.
And by disrupting those essential interactions, we can effectively cripple the parasite's ability to infect us and replicate. And this approach has the added benefit of potentially circumventing drug resistance because the parasite has less control over those host factors, right?
It's brilliant, isn't it?
Yeah.
But let's not forget about the human side of all of this. I mean, malaria disproportionately affects the most vulnerable populations in the world.
It does.
Particularly young children and pregnant women in sub-Saharan Africa. We can't just focus on the science. We also need to address all of the social and economic factors that are at play here, right?
You are absolutely right. Scientific advances alone are not enough. We need to address those social, economic, and environmental factors that contribute to the spread of malaria and that hinder access to healthcare.
Right.
It's about taking this holistic approach
Holistic.
that really tackles the root causes of malaria and empowers communities to protect themselves.
So we need better drugs, We need better vaccines, we need better diagnostics, but we also need better infrastructure, better education, better access to healthcare.
I do.
It's a multi-pronged approach.
It is.
It's a global effort.
Absolutely. It's a fight for global health equity. It's a fight for a world where everybody has the right to live a healthy and fulfilling life, free from the threat of malaria.
Beautifully said. As we wrap up this part of our deep dive, I'm really struck by just how incredibly complex this disease is.
It is complex.
You know, from those really intricate molecular mechanisms of the parasite to the multifaceted challenges of control and prevention, it's clear that malaria is a formidable adversary. But it also highlights just the incredible resilience of the human spirit, and the power of scientific ingenuity. Okay, so deep divers, we've gone from these sticky proteins, this microscopic world, all the way to this global fight against malaria. But I feel like the story doesn't end there, right?
No, definitely not. I mean, there's so much exciting stuff happening on the cutting edge of malaria research. We're talking about innovations that could totally revolutionize the way we approach this disease.
I'm ready. Hit me with some good news. What kind of innovations are we talking about here?
Well, we're seeing innovation on multiple fronts, really. Think about new drug targets, new vaccine strategies, and even artificial intelligence, AI.
Hold on. AI? In malaria research, that sounds pretty futuristic.
It might sound futuristic, but it's already happening. Right now, AI is being used to analyze these massive data sets of information about the parasite. It can see these patterns that we might miss, and it can even help predict drug resistance before it even happens. Imagine that. Personalized treatment plans based on what AI reveals about a patient's specific parasite strain.
So it's like giving doctors a superpowered magnifying glass.
Exactly. And that speed. It's super important. Malaria is such a challenging disease because it adapts and evolves so quickly.
Right.
We need to be able to stay one step ahead, and that's where AI can really help.
Right. So we need all the help we can get in this arms race against malaria. What else is cooking in the research labs right now?
Well, there's a lot of excitement around these new transmission-blocking vaccines.
Transmission blocking, so they're not actually protecting you from getting sick.
Not directly, no. They're stopping the parasite from spreading from an infected person to a mosquito in the first place. So if a mosquito bites someone who's been vaccinated with a transmission-blocking vaccine, that mosquito won't pick up the parasite, and then it can't spread it to other people.
That's incredible. So it's like cutting off the problem at the source.
Exactly. And some researchers are even looking at genetically modifying mosquitoes to make them resistant to the parasite.
Genetically modified mosquitoes, that's next level stuff. What are the chances of that actually working?
I know, it sounds kind of wild, but the research is moving really quickly. And when you think about it, mosquitoes, they're the vehicle. They're spreading this disease. If we can control their populations or just change their ability to transmit the parasite, it could have a massive impact on malaria transmission overall.
It's definitely a bold approach, that's for sure. But the payoff could be huge, potentially.
Wow, this deep dive has been quite the journey, honestly. We started with the sticky protein, and now we're talking about technologies that could reshape the entire fight against malaria. Pretty amazing. It is amazing. And when you take a step back and look at the bigger picture, it really highlights the incredible power of science, right?
Yeah.
From studying these tiny little molecules to developing these large-scale interventions, we're constantly pushing the boundaries of what's possible.
Absolutely. And to all of our listeners out there, if all this science feels a bit overwhelming, remember that every discovery, every new piece of knowledge, it brings us closer to a world without malaria.
That's what it's all about.
Yeah.
And it's not just up to the scientists either. We all have a role to play in this fight. Supporting research, advocating for those policy changes, raising awareness, even just understanding the disease better, it can make a difference.
So what's the one thing you want our listeners to take away from this deep dive? The one thing they should remember?
That malaria is a tough opponent, but it's not unbeatable. The future of malaria control lies in innovative research, collaborative efforts, and just a real commitment to making a difference.
I love that. I couldn't have said it better myself. And on that note, dear listeners, we leave you with this final thought. As we keep pushing forward with all these amazing advancements, how do we make sure that they're used responsibly, equitably? How do we make sure that everyone, everywhere benefits from this progress? That's something to think about.
That is something to think about. It's a conversation we all need to be a part of, a responsibility we all share.
Absolutely. Thanks for joining us for this incredible deep dive. Until next time, stay curious, stay engaged, and let's keep pushing for a healthier world for all.